All right, so my name is Gates. I'm a master's student in Chris's lab. I'm mostly working on sampling design for SCR. So this lecture is going to go in 6 parts. The first is an introduction to the idea of design, and then moving on to comparing classical capture-recapture to spatial capture-recapture. From there, looking at considerations for design in SCR, which is kind of the main point of all this. Going into previous research, and then the new oSCR framework that we're proposing here, and a tutorial on how to use that framework. So introducing the idea of design. It's important here that we talk about this because I think it receives very little recognition and attention, which I note at the bottom of the slide here. But focusing on design allows us to address key considerations, such as, how well will we be able to estimate what we care about, so what parameter that might be, so things like density in particular. Will we get a good estimate? There's some variation there in terms of how precise do you want it to be, or something like that. Will we recognize early on if things are not going well? So if you go out and you have your sampling design out there, you're starting your study, will you know within the first occasion or something if you're getting the data that you would expect? So that's a really key component here, is that design is all about understanding your expected data. And that's a function of prescribed function of the design and not prescribed, which are the parameters that are basically governing the study. So these things may seem obvious that you can sort of play in your favor by, going ahead here. You can play in your favor by thinking about sampling design ahead of time. But at the same time, as I said, it's received relatively little attention. So a few reasons for this, although we just went over this, it still might not be entirely obvious that you can sort of, have some expectation by thinking more about your sampling design. There are only a handful of papers on the topic. So Andy's had some, Katsun has had one on it, Beth Gardner, Rahel Solman, a few others in the SCR community have focused on it a bit, but it could still use some more focus. Most people don't have much of an idea about how to think about or analyze the design problem. So it's a bit backwards because it's based on data that you expect to get, but you have to kind of come up with that beforehand. So it requires a bit of a different way of thinking about it. Importantly, no one wants to commit a bunch of their funding to pay a statistician ahead of time. So oftentimes, you fill out your budget to, get your grant money, and that allows for some fixed number of traps within a study area and some fixed sampling occasion. So you're not really kind of thinking about study design before you get to that point. But I think what we're trying to say here is that maybe that's something that you should think about, and we're providing a tool that should help you understand that. So classical capture-recapture versus SCR. Sampling design in capture-recapture, there's mostly just these two components of your expected data. So that's the number of captures that you get, like the total number of individuals that you capture related to that, and the number of recaptures. Importantly, though, there's little focus on actually where to sample. So there's some key components there. For mark recapture, we think, okay, we don't want any holes in the study design. And then we try to account for individuals on the edge of the trapping grid or stuff like that, and coming up with mean maximum distance moved. But there's not really a formal focus on it. And the pieces that we do think about aren't, entirely encompassing the whole problem. So really the only design element is the number of sample occasions, which is K. And then parameter P is prescribed as the per study period probability of detection. And essentially we want a spatial design that will optimize P. Again, not really a formal basis to account for this though, and we seek to minimize heterogeneity. Compared to SCR though, where to sample is actually the most controllable aspect of sampling. And as we saw in the last slide, it also is the best way to make sure we're getting good estimates. The good thing about SCR, or one of the many good things about SCR, is that it explicitly accounts for space. So it thinks about where individuals live and where we sample them. Those are really important pieces of SCR and important pieces of understanding the spatial ecology of our species. So this is all to say our expected data is mainly influenced by trap placement. So just further motivating that point that we really need to think about trap placement, trap configuration when we're coming up with what our design should look like and thinking about our expected data. So considerations for SCR. We begin with the ICR model and parameters that we need to estimate. So obviously the ICR model is the model we're interested in. Right now we're just going to work with kind of like the most basic SCR model. parameters, typically, total abundance or density, which are related very closely. Two options for the main goal of designing a study. So these are basically two ways that you can think about setting out on thinking about how to set up your study design. The first one isn't used as often, but it's to achieve a target level of precision for estimating parameters. So, how precise do you want your estimates to be? And then you design your whole study based around that, which could mean, you include more cameras or something. Alternatively, that could be a power objective instead of a precision objective, which means the probability of detecting an effect of some magnitude, between two places or something like that. But funding doesn't typically work like this. Most of the time, you're given your budget ahead of time. So you want to maximize your precision for that given level of effort or number of traps. So we were given a fixed budget. Now let's do our best with it. Classical example. This is from one of Andy's old slides. We can afford 70 camera traps and the labor to deploy them, plus sending some grad students to an exotic destination. That's something I haven't quite done yet with Chris, but maybe one day we'll get to there. So if we kind of narrow down this design problem statement to like a single question, it's what spatial configurations of traps maximizes the precision of SCR model parameters? So if we think at a broader scale, what affects statistical precision? First of all, there are things we can't control, right? Basically, the biology of our species described in these SCR terms of your baseline detection rate, space use parameter, which is related to biology to some extent, and then total abundance or density, which is the inference object, so it's unknown. And we talked a lot about the state space and how important the state space is. I think Andy brought it up in his first lecture. Chris talked about it again, and even Dan was talking about it in terms of speeding up your models and stuff. So it's important to think about the state space for sure. But things we can control even before we get to all of that is trap location, which is the design represented here by all the x's, x1, x2, and then duration of sampling. So on a budget, both of those things are fixed quantities. So you have your thing planned out ahead of time for some number of occasions and also 50 traps. Compared to capture-recapture, SCR requires a third consideration. So again, for capture-recapture, it's captures and recaptures. For SCR, those captures are the sample size n, and the recaptures are the number of individual, is directly related to P0, which is the baseline encounter probability. The third one, though, that's really a key point here is the spatial pattern of encounter histories, and that's directly related to space use sigma. It's really helpful in estimating sigma, and from there, it improves your density estimates. That was something that Dan was saying we would talk about here, that it's really important to get good resolution on sigma. In terms of design, though, there's a trade-off between these three elements. In a way, we want to maximize for all of them. We'd love to get a lot of captures, a lot of recaptures, a lot of spatial recaptures, or a good spatial pattern in your encounter histories. But we actually can't really do all of those things. So if you wanted to maximize for captures, you'd have to have your trapping array or grid fill out your entire state space. But if you want to maximize for spatial recaptures, that's a bit more of a constricting design. So you'd be sampling a smaller area, getting fewer individuals, but getting more spatial recaptures on those individuals. Again, assuming that you have a fixed number of traps. So we can't maximize all of these things together, but we can think about these as objectives going into our study. This is what I was describing here. So if you spread traps out, they become, you would want them to be evenly spaced. That should yield a lot of unique individuals within the study area, but probably few, if any, spatial recaptures, depending on the value of sigma relative to the trap spacing. So in a way, you're really maximizing for captures. If you arrange the traps very closely together, we can produce a lot of spatial recaptures, which solves the problem in the first one. But in reality, then we're capturing very few individuals, and we're actually sampling a much smaller area in the study area. So that could also lead to some problems in estimating spatial variation and density, but that's a bit farther down the line. So very few unique individuals, essentially maximizing for spatial recaptures. So here's what that would look like. This is just a quick ggplot. The red circles here represent just theoretical home range areas for a few individuals, and they're the same in both plots. But if you think about it, the light blue here is the state space, and the dark blue crosses represent possible trapping areas. So on the left, if you have your traps all spread out, you can see we capture a lot more individuals in that there are a lot more individuals that are exposed to traps. And then on the right, we see that there are actually fewer individuals exposed to the traps, but each individual has more traps within its home range. So on the left, we could capture 7 individuals on the trapping grid. On the right, we only could capture 3. But on the left, each one's exposed to around just over 1 trap, but on the right, it's exposed to just over 5 traps. So some good things on either side there, we want more individuals, as I was saying, but we also want more spatial recaptures. Getting into previous research, this is from a secret document that Murray Efford put together that sort of formalizes this idea graphically. So we can see that around sigma, which is that vertical dashed line there of 20 meters, that's where we maximize the number of recaptures and also maximize the number of spatial recaptures. But as we increase trap spacing, just keep going on there on the x-axis, we get more and more animals that we capture. That's n, the total sample size. If we look at this in terms of precision, we're maximizing our precision or minimizing it in a way here at around 2 times the sigma estimate. So sigma is 20, and then right around that 40 area is where your precision is maximized. And this has led to a lot of people focusing on the 2 sigma design or 1.5 sigma. So it'll be just a continuous trapping grid and a regular grid, regular area, and each trap is spaced about 2 sigma apart, which is a good first approximation and good things to think about. But there tend to be some problems with that in reality, which we'll get to. Another systematic approach for varying trap spacing is through cluster designs. So there are some pros and cons here. Pros, it allows you to cover more area while simultaneously allowing for greater resolution for estimating sigma. It makes designs that can outcompete standard grid-based designs because essentially you're clustering the traps, spreading them out so you can get spatial recaptures at a fine scale and at a larger scale, but also capture more individuals because your entire trapping extent is a bit wider and longer. On the other hand, though, there lacks a single standardized framework for making one of these designs. So there are a lot of arbitrary decisions that could lead to designs being actually worse than expected. So you could read this paper that says, okay, let's use cluster designs, but if you aren't careful, you could actually come up with a design that doesn't perform as well as you might expect, given that that cluster designs perform better in some cases. And this is because, in effect, a cluster design represents a single realization of the concept. So there's a lot of room for variation. So it's basically just, there's this idea of cluster designs, and each cluster design is just one realization of that. There are 2 components to cluster designs. One is within cluster sampling, and among cluster sampling is the other one. So within cluster sampling provides spatial recaptures that I was talking about, and then among cluster sampling increases the total sample size of individuals. You can also have spatial recaptures among clusters, but that would typically be a bit less common based on your trap spacing. There are several design considerations then, sort of as I'm suggesting here. The allocation of cameras within and between clusters. So that's basically asking the question, how many traps will you have within a cluster, and how many traps are you going to have total, and how many clusters are you going to have altogether? What's the spacing within a cluster between traps, and what's the spacing among the clusters, and how many clusters are there? So this is an idea of what that looks like. So on the bottom, we're essentially increasing the spacing within a cluster. That's on the x-axis, and on the y-axis, we're increasing the spacing between clusters. So if we start in the bottom left there, we see what looks like it could be a reasonable design. You have 9 clusters total. Each cluster has 9 traps. That's a lot of traps. And there's a good variation in spacing between the traps so that you could get good resolution on sigma. If you move to the right from that one, we see that we can just increase that within cluster spacing, which again looks like a fairly reasonable design. And then we can move up to the top one on the left there. And that one, we're increasing the distance among clusters. So you're covering more area there, but you're leaving less room for capture between each cluster. And then moving to the top right, that's sort of a quite different design overall than the bottom left, where you're increasing both within cluster and among cluster spacing, trap spacing. The main conclusion here, though, is that it's a bit tricky to figure out which one of these designs would be best for your study area, your species. And that's actually while maintaining these critical parameters here. Here we have only so many traps. That would be another question, how many traps should you have? And we're also maintaining the same number of traps within a cluster. So you can imagine there's a lot of variation here. And essentially what you'd have to do is take all these designs, simulate some data, recover those estimates using the design and fitting a model. So it gets a bit tricky. You'd have to evaluate precision that way or bias and come up with one. But there should probably be a more straightforward solution, which is what we'll get into. So that's the oSCR framework that I'm presenting here. This is some work that I've done with Chris and Andy and Ali Nawa, who's a snow leopard guy. Some of this has been touched on in what I think is chapter 10 of the oSCR book on sampling design. Andy has a chapter about it there, but we've sort of formalized that process and brought in some new tools to kind of work with it. So where are we? We've seen some good designs and some good ideas related to trap spacing and clustering. We could use these as general rules to create designs manually, and we could evaluate the performance of a design using simulation. That all sounds good. But in reality, as I've been hinting at along the way, there are a lot of questions still. So I've titled this slide, Reality Check, a.k.a. 20 questions. You can see all the red question marks there, that if I were to ask you right now, okay, you have to make this study area or a sampling design for your study area, how would you go about doing that? At first, you might think, okay, like I'll just use a cluster design and it should all work out. But if you really think about it, there are a lot of questions here. Would you choose a regular grid, a cluster? You don't have to think about all these right now, but just to give you the idea that there's a lot of questions. What would trap spacing be? What would that be in a cluster design? Would you make one design or would you test several? Once you choose a design, what can you say about your expected data? And importantly, two really key points here are, what if there are areas in the grid that you can't sample? So within your trapping grid, what if you can't sample some of those areas due to any sort of logistical constraint? That could be, private property or altitude or just slope or anything like that, areas that are hard to get to or impossible to get to. And the other one is, what if the area you're sampling is not rectangular? So again, we can use cluster designs and grid designs, but oftentimes our study area isn't a grid shape or rectangular in any way. So how can we sort of optimize our design to these more biologically relevant areas? And that's something that is really the main focus of this design generation process, is bringing some biological realism to the design process. There's still many decisions to make, and the process is far from unambiguous if you're using these sort of grid-based and cluster-based designs. So our alternative approach, instead we offer a new approach, which is using a design finding solution that starts with a design criterion. And I'll get into the details here along the way. But essentially we want to identify a sensible design criterion that links information content of designs to the sample locations. So this criterion you could think of as like a metric of how good a design is to meet some objective. So an objective such as, increasing your total sample size, number of captures, or increasing your total number of spatial recaptures, or probability of either of those is mostly how we think about it. And step two is we can formally optimize the criterion, again, that metric, over all possible designs using a genetic algorithm. And that's something that we'll get to describe pretty much in detail in the following slides, what the genetic algorithm actually is. So there are some formalities here. We have the design, which is the location of J traps. We have the criterion, which is in our case model-based, so it's derived directly from the oSCR model, and it's related to a parameter of interest. And then importantly, you have your target species parameters. So oSCR parameters that are leveraged by the criterion. So if you have an idea of space use for your species, you can incorporate that to come up with a good design. Our framework is flexible in that it can utilize any oSCR sensible criterion, but the two that we'll start with here and that we've sort of discussed so far are two fundamental criterion that are explicitly leveraging the encounter process in the oSCR model. So QP or QP bar is optimizing for the probability of capture. P bar was something that Andy talked about yesterday and mentioned briefly that we would talk about in this design lecture. And then QP bar M is optimizing for the probability of a capture at more than one trap, which really is a spatial recapture. And it's called QPM M as a movement recapture. So in order to derive these criteria, we start with a standard oSCR model. So this is something we've looked at several times so far. There are two model components, a spatial model of abundance describing the distribution of activity centers, and a spatial model of detection that relates encounter rates to the distance between the activity center and a trap. These should be quite familiar to you at this point. The most basic model form assumes a uniform prior for the distribution of activity centers. That looks like that equation there, where capital S is the state space, discretized into pixels, describing all possible locations of activity centers. That was related to sort of like the trap buffering or the state space buffering that we talked about. And within that region, there are n total individuals observed or exposed to capture, and little n observed individuals, and then little n zero, which is the number of unobserved individuals. So as I said, these two criteria explicitly are derived from the encounter model. So we can use the half normal encounter model, which Chris discussed is pretty robust and a really good choice for oSCR. It describes encounter probability as a decreasing function of distance from an individual's activity center. One thing to note here is that we use G0 instead of P0 when we're denoting the baseline encounter probability. That's just because later on, as you'll see, we use P0 to represent the probability of not being captured when we're deriving the criteria. That's on the next slide, but don't get too caught up on that right now. The main point here is that we have this encounter model that we can use, and all these components should be quite familiar to you. So math folks out there who really like this kind of stuff, I'll kind of show you the math here, but without getting into it in too much detail. So from equation 1, which is the half normal encounter model, we can use values of G0 and sigma, which are species specific, to compute the probability that an individual with an activity center S is detected at any trap in an array X. We can get the marginal probability of not being encountered, which I said earlier was P0. That is kind of, in conflict with the G0 parameter that we're using now. We can take the average over all activity centers, essentially coming up with the marginal probability of encounters. This is P bar. That was what Andy was talking about, what I just mentioned, and that's what one of the criterion is entirely based off of. And then to get the second one, we need to come up with probability of being captured at just one trap. And essentially we can figure out the probability of being captured at more than one trap, or spatial recapture, by doing 1 minus the probability of not being encountered minus the probability of being encountered at one trap. And that's, that'll just get you probability of being encountered at more than one trap, whether that's 2 or 3 or whatever. So then by minimizing these quantities, we arrive at our criteria. So QP bar M is just, you take the negative of P bar M, and for QP bar, you just take the negative of the P bar criteria, or quantity. And essentially this is akin to just minimizing the negative instead of maximizing the positive. We can also create a third criteria by balancing these two objectives explicitly, which is kind of an interesting thing because we're talking about how for oSCR, you need to sort of maximize the number of captures, but also the number of spatial recaptures. And that's something we're starting to try to get at here with the QP bar B criteria. So these are the 3 criteria to keep in mind going forward. So how does the genetic algorithm work? How does it take on these criteria and come up with a design? One of the main points here that I think is really helpful for people is to understand that the genetic algorithm is a computer algorithm that's in the class of evolutionary algorithms. So a lot of the terminology and how it comes up with designs or solutions is based off of evolutionary biology concepts. So sort of as a broad overview, it produces multiple generations of solutions, or that's just sort of like iterations, where subsequent generations retain a characteristic of top performing solutions from the previous generation. And those generations are produced until converging on a near optimal solution. Within each generation, there is a population of solutions, and that's something that we'll get to in the next slide about hyper tuning these sort of algorithms. And then, so the genetic algorithm is a random search algorithm to solve what's known as the K of n design problem, where some number of elements K must be selected from n possible options that minimize an objective function. So that might sound a bit ambiguous, a bit confusing, but in reality, it really concisely describes the challenge of oSCR sampling, where some number of traps K selected from a landscape of n possible trapping locations according to a design criterion. So you can imagine that we have n possible locations where we can trap, but your budget has 50 traps that you have. That's K. So you try to find the 50 positions of n that maximize this criteria or optimize the criteria. So a bit more detail about the design generation process. Importantly, given approximate values of oSCR parameters like baseline detection and space use sigma, these objective functions can be evaluated analytically for any number of configuration of traps. So those values could come from the literature, previously published articles, or from a study design, like a pilot study. We can then apply a genetic algorithm to the task of design finding, which is what we talked about. It's an evolutionary algorithm. It's basically a randomized search process. And that can generate a design optimized according to a selected criterion. We consider designs generated this way to be optimal with respect to a criterion. So they're not optimal in that they can work for any study area, any species, anything like that. It's just that they're optimal with respect to whatever criterion you picked. So are you trying to maximize for number of captures and number of spatial recaptures? And they're optimal in that they've been optimized to find a solution. If you're a visual learner, it's a bit helpful to think of it this way as sort of like a workflow diagram. And this algorithm is adapted directly from an implementation by Wolters in 2015. He has an R function that's called K of NGA. And essentially, what we're proposing here is SCR design GA, which is a wrapper around that original function that adapts it to be used for SCR by incorporating those SCR parameters. So there are 3 parameters to tune the algorithm. Those are pop size, keep best, and NGen. And those fit into the general framework here pretty obviously once you kind of get a handle on it. So it starts off by generating a population of designs. Again, we're using these sort of evolutionary biology terms. And you can tell it how many individuals within that population by parameterizing the pop size. And then you can use keep best to figure out how many within each population generation to keep. Those are scored, and then so all designs are scored, and then you select those top designs, whatever keep best is. From those top designs, those you can breed them together to create new designs, and then the process pretty much starts over again. And that loops through for as many times as you have specified for NGen, which is the number of generations or iterations. And after that, the algorithm is terminated, and it returns the best design. We recommend using at least 1,500 generations in order to run this process smoothly. If you use less than that, the algorithm might not converge on a best solution, which is just important to think about. This is what the function looks like using the algorithm. So it leverages the biology of the study species to find an optimal design while incorporating information on logistical constraints. So what we see here, there are the study area objects that you put in, the species SCR parameters, and design criterion, and then some extra bells and whistles that help with the process along the way. Moving into the tutorial, this is something you can follow along if you want to. You don't necessarily have to. It's pretty straightforward and fairly brief, just because the whole process is made to be quite user-friendly. So the development of this framework was motivated by a large area in northern Pakistan. That was the focus of a snow leopard trapping study, but there are several logistic constraints. So the area that we're interested in is this red polygon on the left. We can see it's in a really mountainous area. Some of those mountains are snow-capped, so you can imagine they're really high, and then it drops into a river valley. So there's some really steep areas there. Along with that, there's some military bases that we have to account for that we can't sample in those. And it's in the Tibetan Plateau. So on the right, you can see Pakistan is outlined in white, and then the little red dot there is essentially the trapping area, and yellow is the snow leopard range, global snow leopard range from the IUCN. So jumping right in the book down website that we have right now that Dan showed, we have some information about how to come up with these spatial objects of the state space and the trapping grid. But we're going to assume that you have those. But if you want more information on that, that's definitely the best place to look because we walk through it step by step. So each of these objects is a data frame with X and Y as the column names. We can see those below. This is what they look like. Again, another ggplot where light blue is the state space and then dark blue are the possible trapping areas. So going back to this whole idea of K of N, so those dark blue points represent the N possible trapping locations. And we can see, importantly, that there are some areas in here, if you can see my cursor, that have been kind of like sliced out from the possible trapping areas. So that might be a military base. Some of the smaller points in here might be because of altitude or slope. And also like this big cutout here is probably due to altitude and slope. So overall, even your possible trapping grid has these holes in it. It's not even that your design has holes in it yet, but just the areas that you can sample, there are holes in that. Including up here at the top, there's a set of just 5 possible locations up there that are entirely, not contiguous with the rest of the area. So we need values of the SCR parameters in order to run this algorithm. So what I'm doing right now is just kind of taking that original R script that I showed you and walking you through what each piece of that looks like. So these values can be obtained, as I said, from literature or a pilot study, and we'll use parameters that from the previous section. So we have your baseline encounter probability, that's 0.2, number of sampling occasions, which is K or 5. And then we use the detection intercept, which is just the log of those 2 detection parameters above, and then sigma, which is 2.59. And then finally, we have 50 traps that we want to optimize our design with. With everything in place, we can run the design finding algorithm. So it just, everything plugs in like that. And basically, it's essentially plug and play. It should be a really straightforward process. The main footwork is figuring out these parameters ahead of time and also deciding on a design criterion. So here we see crit equals 2, and crit is the design criterion. 2 represents the QPM criterion. 1, if you set crit equals 1, that's the QP criterion, and then if crit equals 3, then it's the QPB criterion. So once we run the design, we've run it here for only 50 generations, which should give you an idea that right now this is probably not going to converge at this point. We can plot these diagnostic plots for our design. So from left to right, these show the value of the objective function by generation, and then the optimized design output and a histogram of trap spacing. So that's what you're seeing here. Plot.scr design just takes the design object, the output of the algorithm, and shows you these diagnostic plots. The one on the left there is the one that's probably of most interest if you want to make sure that your design has converged, or the algorithm has converged on a design. So on the y-axis, we have the objective function value, and on the x-axis, we have the generation. So we ran it for 50 generations. That's why the generations go up to 50 on the x-axis. And importantly, the red line in particular is the one to look at. That's showing the best solution for each generation or after each generation. And we can see generally that that's declining right now, but it hasn't really bottomed out. It hasn't really like minimized or anything at any point. So that's suggesting it hasn't converged. And we can also see that in the middle plot there, which is the design output. And those traps look like they have sort of like a general design shape to them. And you'll get a bit more of an intuition for this once you use the algorithm more. But they look a bit scattered still, which is to be expected since there's only 50 traps here, and it's only 50 generations. And then the one on the right is it's a histogram of the distribution of trap spacing, which isn't as informative about convergence, but it is informative about how your design works, how it will operate, especially relative to sigma. And we can see the value for sigma is that vertical red solid line in that figure. And then right next to that, there's a dashed vertical line that represents 2 sigma. The blue histogram bar right there represents sort of like the dominant trap spacing between nearest traps. And then the red bars represent sort of like a secondary trap spacing, which is among all pairs of traps. We can see there's a good distribution there. We can get some good resolution on sigma. I'm going to pause for a second. It looks like there's some questions. Is there anything that I should address here? Chris, Andy, any thoughts?
No, no, I think it's good. There's just a couple questions in the Slack that I think are taken care of already. So.
Okay, great. Sounds good. Okay, so to describe these plots in more detail, this is really just a slide for anyone who's following along after the lecture. But I've described all these pieces in terms of, blue line represents population average, red line represents the lowest or the design output after each generation. The optimized design output should be familiar. The design is in blue and going on from there. But the primary thing you want to look for is that the algorithm has converged on a solution. In order to do that, again, we want to see that red line has flattened out, and that should flatten out for at least a couple hundred generations just to make sure that it's stable at the minimum that it's found. And then we can check the design structure. So with each criteria that we've presented here, we have like a general understanding of what designs might look like. So QP bar is a space filling design. So those traps should fill the state space or the trapping grid, and also, subsequently part of the state space. And most of those traps should be really evenly spaced. So there should be like a regular distance between all the traps. QP bar M is space restricting. So those traps will be much closer together. They're not filling the entire area. They're focusing on getting spatial recaptures by bringing all the traps closer together. And then QPB is clustered space filling. So it's a lot like QP bar and what it will look like overall. But if you look more closely, it ends up having a lot of traps that are sort of paired. So you have some small clusters of traps throughout the state space. That should help with spatial recaptures, but still the trapping grid overall is spread out so that you capture a lot of unique individuals. If it looks like the algorithm hasn't converged, we can simply just increase the number of generations that we run the genetic algorithm for. So we recommend, again, above 1,500 generations. And an easy way to do this, or a really efficient way to do this, is we can actually supply the genetic algorithm with a cluster a cluster of cores on your computer, not like a cluster design. And that should significantly decrease your runtime. So these designs should converge much faster. This is the code to do that. I wouldn't recommend trying to do this right now. Your computer, your fan will start going really fast and everything like that. But for the future, if you want to sit down and actually really think about making a design, it's definitely a good way to go is to introduce this cluster. So we have the cluster set up here where we detect the number of cores available on your computer. We subtract 1 just to leave 1 core available to your computer in case it needs any sort of troubleshooting or needs to take care of other tasks automatically. We make that cluster by supplying the number of cores that we want to put in the cluster. We export one of a critical function to the algorithm, to the clusters. And then we just start running the design by supplying that cluster right to the function there with cluster equals CL. That will run, it should spit out every time with verbose equals true, it should spit out which generation it's on. So let's say, generation 1, generation 2, it'll spit out the value of the criterion as well for each generation. But essentially, once you supply it that cluster, that output should start running off really fast. Once it's done, just make sure that you stop the cluster. If you don't do that, it doesn't cause any huge problems, but it slows your computer down quite a bit until you realize that you have to do it. And it might spit out some weird errors along the way. So just make sure you stop it. So this is what the design will look like if you've run it for long enough that it's converged on a solution. So what we're seeing there is that red line has a really nice shape to it. It drops down and finds the minimum, and it stays at that minimum value for about 200 or 300 generations there at around the 1200 mark. So on the middle plot, we see that the design has much more of like a structural characteristic to it. Often this QPM criterion can be described as having like a ring of traps in some sense, which we're kind of seeing here, and then maybe some traps in the middle, which allow for some resolution on sigma. So that looks like a pretty solid design. One thing that you might notice though is that we're not actually sampling the entire study area using that design. And that's sort of one downside of not having enough traps really. So that's not really an issue of this algorithm or our process or anything, but it's really just the fact that with less sampling effort, you get to cover less area. That's related to what Andy was talking about with the effective sampling area, ESA. If we use more traps, we can increase our ESA and we can get some better data from that. And on the right, again, just the same histogram plot that we talked about before. Again, not super informative about convergence, but still interesting to look at and to help you understand exactly how your design will work. So once we have these designs, obviously they've been optimized, so they should be good designs, but it's always good to go through how to simulate data and then try to recover parameter estimates by using one of these designs. And that can tell you, how good your design is and what sort of data you can expect to get out of it. So we illustrate this using the simulator function, which is available on the book down. This executes the following steps here, steps 1 through 5. Essentially, it's just simulating oSCR data, which I think we've talked about before anyway, but it randomly assigns n activity centers to the state space. It calculates detection based on the distance between an activity center and each trap. It creates a 3D encounter history using the binomial model. And then again, that's coin flipping. Fits a basic oSCR model using oSCR.fit, and then it returns parameter estimates and summary statistics. So running the simulations is pretty straightforward with simulator, much like the genetic algorithm function. It's really just plug and play. So you can put in your trap locations, your state space, and then all those oSCR parameters. And importantly, the oSCR parameters that you use for the simulation should be the same that you use to generate the design, because you want to make sure that the design is optimal for these parameters. So you're simulating using the same ones that you generated the design with. And we just plug that all into simulator. If we use plot equals true, then that should plot each simulation along the way, which is a nice just visual check with the spider plot function. And then we recommend using at least 50 simulations to come up with what your parameters will look like or your estimates will look like. But really more than that is always better. So in our design paper that we have on bioRxiv right now and in review, we used 300 simulations, which seems like a good spot, kind of the sweet spot between not running too many simulations, but also running definitely enough that we have a good understanding of what these estimates will look like. So once we have the estimates, we need to compare those to the original values of the parameters. One pretty immediately interpretable way to do this is by using percent relative bias. That's been used in other design papers. The equation shown there, pretty straightforward. Other useful metrics, though, can include coefficient of variation if you want to look at precision, scaled root mean squared error for accuracy or coverage. But percent relative bias is good because if you do it per each simulation, you can actually get a sense for precision as well. And then on the bottom here, with the tidyverse, I'm just kind of showing how you can calculate percent relative bias. But don't worry too much about that. It's pretty straightforward. Once we get that, we can plot these again using ggplot. So we have each parameter on the bottom there, p0, sigma, and n, and then percent relative bias. And those are all represented by box plots. The horizontal solid gray line is showing zero bias. So that's when you recover an estimate that's the same as the parameter that you simulated with. And then the gray dashed lines that are horizontal represent plus or minus 5% bias. So that's like an allowable amount of bias. We're okay if our designs have that much bias in it. And we can see across the board here, on average, the design performs very well. Everything's within that allowable amount of bias. Most of it's right on the money. Importantly, sigma is estimated quite well here. And that's pretty much to be expected because we use the QPM criterion. So it's maximizing for probability of a spatial recapture. So it should be getting good resolution on sigma by getting a lot of recaptures. And then we have a good overall abundance estimate there too. So we're just wrapping up here to get to the take-home points. In general, where to sample depends on the oSCR model. So this is a problem of optimal design, according to that model. Trap spacing is an important consideration, but it's inconclusive. Again, think back to that slide where we had the 20 questions. How many traps should there be? What should that spacing be? Should it be a grid? Should it be a cluster? Cluster sampling is efficient and generally unbiased. It can be better than just regular grid sampling, but that assumes that sampling is adequate, which is a bit hard to judge. And the design generation process overall can be quite ambiguous. Again, think back to those questions about spacing within a cluster, spacing among clusters. The framework that we propose here for oSCR, it's a conceptual and analytical framework for generating optimized designs. We propose 3 intuitive and statistically grounded design criterion. Again, those are based directly on the encounter model. So they're leveraging the encounter process within oSCR. They produce a greater amount of expected information, leading to more accurate estimates. And they're flexible for application to any species in any study area. So as you saw along the way, we can use any state space, any possible trapping grid, and any species in that, any of those oSCR parameters for a species. So it's really flexible. It should be really useful for everyone.